Good morning, Dr. Wimmers. Um, this is Jared Arrington from Bio 200 Lab um, at Towson University, obviously. And this is the Enzyme Lab presentation. It's pretty early, pretty tired, but we can get through this. Um, so, the main thing with this, um, with this exact experiment was to try and test, a certain enzyme and try to recreate um, you know, an experiment um, that can allow trypsin, an enzyme, to and see how well it can break down um, objects. Um, and so, first, some background: trypsin is a proteolytic enzyme that digests dietary proteins. Um, Basically, it's found in the small intestine. Um, so it's a dietary, it's something that helps break down food and stuff like that. Get our waste and stuff like that. Trypsin is also known for being one of the several proteolytic enzymes that degrade it too. Trypsin works specifically by catalyzing the hydrolysis of the peptide bonds in the matter. Um, and trypsin's enzyme activity is more specific to the carbonyl side of the amino acids lysine and arginine. Um, the main reason um, that we chose trypsin in the way we conducted our experiments is that trypsin is active in the stomach um, and where the optimal pHs and temperature of that is the pH is around like 8 and the optimal temperature is around 37 degrees Celsius. And so we wanted to try and recreate that with this experiment specifically. Um, and so one of our method was that we chose 4 different pHs at 5, 7, 9, and 11. Um, and we did uh, 3 trials at each, and then we did 3 trials um, for our negative control at pH 8, where we didn't add any trypsin. We just added more reaction buffer, um, which was the azocasein. Um, and so here are our results. I don't know if you can really see that. I don't know if you can really see that. But those are our results. Um, but um, our results basically stated that the optimal was around 7. It was a very, very quick drop off from 7 to 9, um, which was kind of weird because when you look at other people's data that did the same thing, um, their optimal pHs were around 8, 9, some were even around 10, and so it was kind of weird, to see ours was at 7, and ours was kind of the only one that was at 7. Um, but I feel like our data was correct, um, and as you can see, um, it had a really, really steep drop off, and a steep come up as well. Instead of it being, just from like gradually from like 5, then the 6, then the 7, then the 8, then the 9. It was like 5, like 7 is like the super high, and then it just went straight down. And um, you know, based off of you know, doing my own research, I could have probably assumed that um, you know, and our hypothesis wasn't really supported. Our hypothesis was that um, at higher pHs, the higher pH can get. So as it increases, enzyme activity will also increase. Um, and that was due, me and my group, we decided, that based off of, we thought that as more, um, as more basic stuff can get, then more enzyme activity is occurring within the, within that substance. So our hypothesis was not supported by this. Um, our hypothesis was kind of, and we really should have looked better at other people's data because then we would have been able to see, um, we would have been able to see the fact that everyone else was getting around like pH 8, and then we could have probably changed it, um, you know. But our experiment was adequate. I feel as though. Um, the standard deviation and the t-test results were relatively similar to each other. They all were compared to pH 7. So pH 5 was compared to pH 7, 9 to 7, and 11 to 7. Um, so they were relatively low. There were no significant differences. There were no significant differences. Um, as well as in the standard deviation, it hovered around 0.03, which means it's uniformity in each, which kind of eliminates the thought that we like did something wrong throughout the test, throughout our test. Um, and so, yeah, our hypothesis was adequate. If we were to do this again, we would definitely change our hypothesis based on other people's research. Um, like in specific, I mean, like one group did like pH like 7, 7.5, 8, 8.5, 9, and then that, you can really tell based off of that, that it was relatively, you know, uniform, that everyone kind of understood that it was around 8. Um, that would be one thing. And another thing that we would do different is that we would do, instead of doing 3 negative controls at pH 8, we would do a negative control at each pH. So instead of doing all of 3 at pH 8, we do 1 at pH 5, 1 at pH 7, 1 at pH 9, and 1 at pH 11. Um, and I think that would be more suitable to test our results, instead of just, shooting a blank in the dark and saying, I think it's this. So yeah, that's probably the biggest thing. It really is just looking at other people's work. You know, we didn't really have much human error. I don't think so. If we just looked at other people's work, then we would have been able to see that we needed to get a certain point. We needed to we need to probably like cut it off at like 8, do like 8, 9, 10. Which the group did. So um, yeah. So that's it for this presentation. It was fun. I really enjoyed it. Um, so yeah.